Hi, and welcome to the Restorative Wellness Clinicians Corner, a video series exclusively for functional health professionals, where we interview the top experts in the latest research, products, tools, and best practices for getting your clients exceptional results. So welcome everybody to today's episode of the RWS Clinicians Corner. I am super excited for our special guests today. We have Annalise Smith and Nikki Grant here from 3x4. Do you guys want to say hi so folks can see you in your cameras?
Hi, everybody. Hello. Hi.
We're going to have an incredible talk today on the interplay of genetics and autoimmunity. And so both Annalie and Nikki are from our absolute favorite lab for testing genetics, which is 3x4. When we were doing the research for our clinical mastery course, we looked at many different options for what lab to partner with for our course. And we had a long list of criteria, which I'm not going to bore you with right now, but what I will tell you is that the resounding winner was 3x4. We absolutely love them. We love their interface. We love how user-friendly their report is and non-intimidating for our clients, but also the ability to go dig as deep as you want to as a clinician, how current they're staying with the research. They are fantastic. This is going to be a wonderful talk. So I'm going to read you their bios so you know the incredible expertise we have in the house, and then I'm going to hand it over to Annalie and Nikki. So Annalie heads up the clinical and mentorship team at 3x4 Genetics. She is an accomplished certified clinician who spent more than 25 years in clinical practice and the wellness industry, gaining experience in many different applications of clinical and functional nutrition. She is our people, both of these two, they're our people. This includes highly specialized nutrition interventions for critically ill patients, wellness and resilience support for executives, and enhancing sports performance for elite athletes and sports teams. Annalie has been involved in the field of nutrigenomics for more than 15 years, and she has translated thousands of genetic reports into clinical practice. This led her to being one of the most recognized international experts in the field today. She's been a valuable part of the 3x4 Genetics company journey from the beginning, and now mentors and supports the clinical team and the company's business partners. Her work at 3x4 Genetics includes consulting for elite sports teams such as LAFC, high-profile practitioners, and famous celebrities. Her personal goal is to influence as many people as possible to make meaningful nutrition and lifestyle choices to obtain quality of life. In her free time, she enjoys wild, long-distance cold water swimming in oceans, rivers, and lakes. This is a metaphor for her personal ambition to push the limits past her comfort zone and to go the extra mile. Annalie, welcome. So excited to have you here.
Thank you.
And Nikki Grant is a registered dietitian with a BSc Diet from the University of Natal, South Africa. She has been in private practice for almost 25 years. In 2014, frustrated by her own health journey, she completed the certificate course in integrative medicine offered by the Department of Family Medicine at Stellenbosch University, South Africa. After which she changed her focus and practice to helping patients using a more holistic approach to disease management through addressing root cause rather than symptom management. Her passion within the functional nutrition space remains with autoimmune disorders and thyroid disease. That's very special to my heart as well. In 2019, she began consulting to 3x4 Genetics on nutrigenomics. Nikki completed the Manuka Sciences course on translational nutrigenomics at the end of 2020. She continues to consult for 3x4 Genetics and forms a valuable part of both their clinical and mentorship teams. Nikki is passionate about making complex scientific information understandable to the average person. She is constantly on a learning journey, and in 2022, she became an IFNA certified practitioner. She has presented many talks to various audiences, including medical professionals and the lay public. She's appeared on many radio and television talk shows, as well as written many articles for print media and published a recipe book for insulin resistance management in 2002, The Ultimate Diet Solution Cookbook. cookbook. Having grown up in a family with a brother who had a hereditary genetic disorder, she has always been fascinated by genetics and strives to stay abreast of this dynamic field of nutrigenomics. Welcome, Nikki.
Thank you so much.
With that, I'm going to turn it over to you two and take it from here.
Fantastic. Thank you, everybody, and thank you, Margaret, for having us this evening, or this evening for us, this morning for you. Right, I'm going to go ahead and share my screen. If you could just let me know that I am sharing the correct screen. Awesome.
Yeah,
okay,
yes,
so I get the privilege of starting this duo out. And as Margaret mentioned, autoimmunity is something that is very close to my heart. And I want to remind you that autoimmunity is actually so complex that we could never cover all of its intricacies in this hour. In this presentation, we will be skimming over some of the components associated with the development of autoimmune disease, asking some questions, prompting some thoughts, and giving you a broad overview of the numerous contributing factors associated with the development of an autoimmune condition. And still, we will have just been scratching the surface. The more I learn about this complex group of conditions, the more I realize we have so much more to learn. I'd like to begin by sharing a couple of poems written by people who live with autoimmunity every day, in the hopes that I can help you to wear their shoes just for a little while, at least emotionally. She was tired. No one could see the level of tired. They saw the outside, the one giving, smiling, showing up, and yet inside she felt the fatigue, the tired of trying to keep up, the tired of agendas, the tired of worries, but she kept on. She kept giving and loving. She kept on hoping. She kept on showing up. She knew her giving mattered. She pushed forward out of love, even in the tired. You may not see it, but I can definitely feel it. You don't understand it, but it's what I know. I deal with it on a daily basis. Either you think I'm lazy or feel sorry for me, but I'm stronger than you'll ever know. I fight every day. I don't want your sympathy. I just want to be treated with respect, and I want to feel better. I've forgotten what that feels like. Many autoimmune patients will go on to have 3 diagnosed by the time they die if we don't address root causes. So how and why does autoimmunity happen in some and not in others? It is a complex interaction of the environment, genetics, and immunity, and we're going to unpack each of these. Starting with epigenetics or your environment, remember we need to think about things like pollutants, toxins, hormonal changes, immune-reactive dietary proteins, infections. Often we hear autoimmunity comes out of Epstein-Barr or cytomegalovirus or stress or trauma. Starting with pollution, we just have to look around us and we see the world is definitely the left-hand side of this picture more than it is the right. We are surrounded by pollution. We are surrounded by toxins. We ingest them, we eat them daily without even knowing it. This study provides the first data that thermal paper may be a significant factor in accounting for high levels of bioactive BPA in human serum and a total BPA in urine that have been associated with diseases that are increasing in frequency in human populations. The findings also suggest that the impact of the use of dermal penetrating enhancing chemicals in skincare products on transdermal absorption of environmental contaminants should be taken into consideration. So should we be using those advanced creams that increase the transfer of particles for our vanity? I found this article incredibly interesting especially in light of coming out of the pandemic and how much we were using things like hand sanitizers. And this particular study was published in 2014, a study a little bit before its time in terms of the use of hand sanitizers at the extreme, or to the level that we did in the pandemic times. Just think about how often we went to grocery stores and we took those till slips or cash register receipts willingly, and then plied our hands with these chemicals, forcing that BPA potentially into our systems. Stress and trauma, one of the risk factors for autoimmunity is early childhood trauma, known as ACEs, or adverse childhood events. This diagram shows how blood-brain barrier disruption occurs. So, and its influence on these traumas and the ripple effect of traumas on health. So advances in neuroscience, genetics, and biomarker studies are changing how we view the impact of adversity. When an individual feels threatened, the human body activates different biological pathways designed to protect the individual from these events in order to survive. The initial sympathetic response we know as the fight or flight reaction, produces increased levels of adrenaline and noradrenaline that cause an increase in heart rate, peripheral vasoconstriction, and massive energy mobilization. The hypothalamus pituitary adrenal axis, one of the center stress response systems, provides a protracted response triggering the secretion of corticotropin-releasing hormone. Studies have demonstrated that corticotropin-releasing hormone has pro-inflammatory properties that can regulate the blood-brain barrier leakage. Animal studies of early stress, particularly in rats, so remember that this we can't 100% extrapolate to humans because it is a rat study, have been shown to induce blood-brain barrier breakdown. Several animal models of PTSD have demonstrated how rats confronted with a traumatic event have increased values of this blood-brain barrier disruption biomarkers. You can see some of the inflammatory cytokines and immunologic responses, autonomic responses that are happening within our body, out of our control. This is a biological inbuilt mechanism in response to these stresses. Autoimmune conditions are more prevalent in women than in men, and it has been thought that hormonal differences are the key role players in the development of autoimmunity. When we consider hormones in the context of epigenetics and the environment, we need to keep front of mind the contribution that endocrine disruptors may play. So what are endocrine disruptors? They are any exogenous compound that may interfere with the synthesis, secretion, transport, metabolism, receptor binding, or elimination of endogenous hormones, altering the endocrine and homeostatic systems. The endocrine system is not a set of insular organs, but rather an intricate network of signaling molecules that interact with each other to coordinate the functions of the body. Affecting one endocrine organ always spills over into the other systems because they are so interrelated. So what are these endocrine disruptors? They are found in the air that we breathe, the food that we eat, the water we drink, and the soil from which our foods are grown, which is contaminated. Every person tested in the US has endocrine disrupting chemical metabolites in their urine. In 2005, the Environmental Working Group found an average of 278 chemicals in the cord blood of newborn babies. Unfortunately, many of these endocrine disrupting chemicals cross the placenta freely and have been found in amniotic fluid in the same concentration as in the maternal bloodstream. The dangers associated with the complex mixture of chemicals are also not completely understood or fully understood. Currently, there are approximately 1,000 known endocrine disrupting chemicals. So how do they work? Endocrine disrupting chemicals, or EDCs, fit into the receptors and alter signaling. There are three main effects that result from this receptor binding. Firstly, it can increase hormone activity, so increase estrogen effects. Secondly, it could block hormone receptors, reducing function. So hormone levels in the blood feed back to the brain that there's not enough hormone in the system, while in fact there's insufficient signaling. And the third is the effects are much more than receptor blocking, as endocrine disrupting chemicals may modify genetic expression through DNA methylation, histone modification, regulatory RNA modification, preventing transcription factors from expressing, enzyme pathways in steroid biosynthesis and metabolism, and other pathways. Dose response is not constant. So you may get a higher response from a lower dose, or it may be different effects in males than in females, and also affect different organs differently. This makes it really complex to study. The most studied effects of endocrine disrupting chemicals are on the reproductive and endocrine health itself, brain and cognitive health, and pulmonary and cardiovascular effects. I'm going to focus on the endocrine effects for the scope of this particular presentation. Women are affected throughout their life cycle, with the most sensitive window of exposure being in utero. Genetic modifications here have lifelong influences. This is considered a critical window of exposure due to pregnancy being a time of rapid cell division. Since the first months postpartum are also critical for neurological development of the baby, it too is a very key vulnerable time. And we know that breast milk can also be high in these endocrine disrupting chemicals. A major stumbling block associated with studying the impact of these endocrine disrupting chemicals is that their effects lag, and their effects are often transgenerational. So in this diagram, you can see that the eggs developing inside the fetus, therefore the grandchildren of the current pregnant mother in my little diagram, are affected by these endocrine disrupting chemicals. The lag. So what is the lag? The infant, the baby in utero, needs to reach puberty to determine if there are any hormonal disruptions because of the exposure to these EDCs in utero. Reproductive tissue may be more likely to appear in reproductive issues may be more likely to appear in the third generation. So the egg of the current fetus in utero in this picture, increasing the waiting period. So studies have found that delayed menstrual regularization, higher odds of irregular menstrual periods, and fewer live births amongst women whose grandmothers were exposed to certain medications during pregnancy. Birth defects as well as attention deficit disorder have also been found in grandchildren of women exposed to some of these medications. Because endocrine disrupting chemicals are ubiquitous, the high prevalence of exposure and the possibility of cumulative consequences must also be considered. Phthalates have been shown to alter third generation behavior and stress response, corticosteroid levels, pituitary gene expression and behavior. This again was a mouse study. The BPA that we looked at earlier has been reported to change third to fifth generation social interactions. The first trimester is a critical exposure window, which we mentioned earlier, due to the genome demethylation and de novo remethylation, all these complicated genetic terms. So hopefully you've heard them before. If you're part of 3x4, you definitely have. And the establishment of imprints, which determine sex. So these could be some of the reasons why it is more around females that we see some of these negatives as a result of the exposure to these endocrine disrupting chemicals in utero. So as we've mentioned, autoimmune disease very often seen more often or more frequently in women, but often autoimmune disease is diagnosed after a pregnancy. Mine happened to be, so this is my experience. This has contributed to the thinking around the gender differential of autoimmunity prevalence in women. But new evidence has been emerging in the literature around the concept of microchimerism being a contributing factor to the development of autoimmunity, especially postpartum. So what is microchimerism? This is a bidirectional exchange of fetal and maternal cells during pregnancy. So during pregnancy, fetal cells, represented here in this diagram as the orange and the little green dots, move into the maternal body, increasing in quantity throughout the gestational period. Likewise, each fetus inherits some of mom's cells, represented in the purple dots. It has been depicted that younger siblings could also obtain older siblings' cells, as depicted with offspring 1, orange circulating, within the younger sibling's body of offspring 2 in our diagram on your screen. There are benefits to this phenomenon, and in this picture you can see the payoffs of the so-called mother-offspring tug-of-war. This theory, known as the cooperation and conflict theory, can explain the paradoxical role of fetal cell microchimerism plays in maternal health. The theory hypothesizes that tissues involved in resource allocation, such as the brain, the thyroid, and the breast, would likely be reservoirs for fetal cells, depicted again here in the orange dots or circles. Additionally, the maternal immune system is likely to play an active role in fetal-maternal interactions. Within these tissues, the outcome of these fetal-maternal negotiations, or the tug-of-war as it's termed, are important for both maternal health and well-being, as well as provision of nutrients to the baby. These fetal hematopoietic cells, or stem cells, can be detected in the circulation of women up to 27 years after delivery. Examples of autoimmune diseases where fetal Y chromosomes were detected decades after pregnancy include things like Hashimoto's thyroiditis, systemic lupus erythematosus or SLE, Sjögren's syndrome. Male patients in these studies are usually used as the control. Fetal cell presence in the thyroid should be associated with higher maternal body temperature, and the presence of fetal cells in the thyroid may also contribute to thyroid cancer risk or susceptibility to other thyroid diseases if these cells are proliferating or producing factors that may induce abnormal thyroid physiology. Fetal cells have been found more frequently in the blood and thyroid tissue of women with Hashimoto's thyroiditis, Graves' disease, as well as cancer. I'm definitely going to be watching this space in terms of the research and what its role is in terms of contributing to autoimmune disease, as well as its role in terms of protection of both mom and baby in pregnancy. Then we get to immune reactive dietary proteins, and I'm sure if you are a nutritionist or a dietitian background, the kinds of proteins we're talking about are those ones that are found in dairy, the typical allergens in fish, other animal proteins, gluten, other animal proteins such as eggs, gluten, as well as we're going to have a look at how some of these chemicals become reactive proteins later. Okay, so how can we reduce it? We need to reduce our toxin exposure by eating organic, eating fresh or frozen vegetables, trying not to use microwaves, especially with plastic containers, using glass instead, avoiding cans that are lined with plastic liners, not handling thermal receipts. The list is endless. And again, as I know the audience is mostly in the nutrition space, you don't need to be reminded of these things. My last note on this slide before we move to the next one is read labels carefully. If only labels were so honest. While infections like viruses, bacteria, Epstein-Barr, cytomegalovirus are commonly associated with the development of autoimmune disease, the scope of this presentation doesn't really allow us to dive too deeply into how. Commonly in functional medicine practices, infective agents may contribute to the development of autoimmune disease as a result of what is called the bystander effect. So the example that I like to use here is the chickenpox virus. You get chickenpox as a child, you make an antibody to chickenpox, but that virus lays dormant inside the cell. When you're a little run down, it might manifest in the form of something like shingles or cold sores or something like that. That antibody is trained to kill that virus. So if there is a infection happening as a result of a compromised immune system or just being run down or low, those antibodies could start attacking that virus. And the bystander effect is the collateral damage of cells in that same facility. Just a reminder of our immune system. So we have an innate immune system. Remember that that's the first responder. That's the one that's, first we've got the barriers. So our barriers are our skin, our lungs, our gastrointestinal tract, and then we've got the first line of defense, which are not cells that are programmed for any specifics, but they respond to an invasion. And then we get the adaptive immune system, which now recognizes that that chickenpox virus or herpes virus, and has a code, has an antibody made up so that every time it sees that particular structure, it's going to, or that protein, it's going to attack it. The gut, we saw from the previous slide, is one of our major barriers, okay? It's the major absorption organ, but is also a barrier to the outside world. The loss of barrier integrity, recognized in gastroenterology journals as gastrointestinal hyperpermeability. In functional medicine space, we may speak about leaky gut, and that gastrointestinal hyperpermeability due to zonulin imbalances, stress, chronic medication use, poor diets, disturbed or non-existent microbiomes, low stomach acid, all lead to poor digestion, and both undigested foods, so whole proteins, as well as chemicals breaching that border. I want you to just keep that thought in mind as we run through my immune system military analogy, which is coming up right now. So before we can get into the pathophysiology potentially associated with autoimmunity, I'd like to run through an analogy of normal immune physiology. So an immunity refresher. Okay, so my analogy is a military invasion. Let's assume you are a country with borders and fences, barriers to foreign infiltrations. In this analogy, your skin, your gut mucosa, your lungs, they're the barriers. If the borders were to have dilapidated immigration posts and breaks in the fences, we could have a situation of unwanted invasion. So too, if the barriers of the body are compromised, undigested food particles, bacteria, parasites, molds, environmental toxins, they gain access. This invasion triggers a well-organized immune response. So first on the scene is the macrophages and mast cells, stationed at the barriers, poised to attack and sound the alarm. Imagine him to be a really overweight security guard or border control guard, carrying just a club, not a gun, but he's shouting really loud. Okay, he's going to engulf the terrorist. The macrophage creates an antigen-presenting cell that acts like an air raid siren, calling in the rest of the immune system to come and help. The first to respond to this alarm are the T helper cells, dispatchers, foot soldiers, army, who organize the attack, okay? They send messages to bring in more help, like the Marines and the Special Forces and the snipers. Cytotoxic killer cells or natural killer cells would be those Special Forces. They swarm the terrorist to kill, destroy, and neutralize it. Back at the command center, military generals known as T regulatory cells monitor the situation and ensure that there are enough T helper cells and T suppressor cells, which could be your conscious objectors, as it were, that halt the immune reaction once the terrorist is neutralized. The immune system takes no chances at another attack. Assuming that the terrorist is part of an organized military group, the T helper cells fetch the FBI detectives or your B cells, which attach themselves to the terrorist and download all of its information into its memory banks. This will result in a more efficient processing by the cytotoxic T cells or Special Forces should that terrorist group invade again. The B cells, who are briefed on the invasion, become the spotters for future invasion. Together with the primed cytotoxic T cells, they become like an elite SWAT team for future intruders. So why the refresher in immunity? Well, it's because the definitions of autoimmunity imply that there's a failure in the immune system to protect us. My thought is that it's not an immune system failure, but rather that it is stimulated through a barrier breach. Think about our environmental toxins. Think about our foods that carry all these chemicals and this breakdown of this gut barrier. The rates of autoimmune diagnoses have gone up by 20% in the last 30 years, and in some countries surpass the rates of cancer diagnoses. It is proposed, that autoimmunity, much like insulin resistance to type 2 diabetes, is a progression. And the progression starts with silent autoimmunity, where there's elevated antibodies, but no symptoms, no tissue damage. This is usually seen in the mainstream medical world and ignored. Autoimmune reactivity, we see elevated antibodies with symptoms, but no clinically noticeable tissue damage, okay? Immune disease is diagnosed when the antibodies are elevated with symptoms and measurable tissue loss. Do you know that in Addison's disease, 90% of adrenal gland function needs to be lost in order for the symptoms and measurable tissue loss to equate to a diagnosis, the progression to which is thought to be adrenal fatigue. Stopping it in its tracks, really important. So this is a tool that we developed at 3x4, just walking you through this progression to autoimmunity. And if you are familiar with our blueprints, Annalie is going to actually walk you through this a little bit more comprehensively when she does her case study. But here we've got the pathways that you would want to look out for in the different stages. And to the right-hand side, remember that the gut is a barrier. The gut is really important. This takes us to the concept of cross-reactivity. This is not unfamiliar to us. If you're an RD or a dietitian, I qualified many moons ago, and I was taught about the cross-reactivity between latex and avocado and kiwi fruit. So we know this, okay? These are just some graphics showing you some of the chemical or the foods and environmental, like birch is obviously an environmental pollen, and what it would cross-react to and the risk associated with that in the little dial on the right-hand side. So this is not an unfamiliar concept. Since antibodies are not made to a single amino acid, but to an amino acid sequence, which we call a protein, this is part of the explanation for the loss of self-tolerance. Whole proteins with similar sequences to our own body tissue can result in self-destruction. This slide shows the work of Arista Vajani in 2009, sequencing the proteins of gliadin, the protein fraction of gluten, and cerebellar tissue. The sequences have remarkable similarities. Hence, if a whole intact gliadin protein breaches the gut barrier and antibody is formed to that particular gliadin, there is a risk that that very same antibody could view cerebellar tissue as an intruder. Similarly, streptococcus and myelin basic protein found in our nerves. This particular picture, also some of Vajani's work, shows human aquaporins, which we know are water channels, found in many cells in our bodies, including stomach, brain, lung, and skeletal tissues. And what foods have very similar protein sequences. This particular study was done around multiple sclerosis and the foods that are associated with potentially damaging the blood-brain barrier in that particular autoimmune condition. Cross-reactivity explains molecular mimicry, where the body sees something that is self as an invader and loses tolerance to self by destroying that tissue. Not a failed immune system, but a military-trained precision immune system, but something has gotten in, has breached a barrier incorrectly or inappropriately. This diagram shows some research from Datis Karazian's work in 2014 and shows proposed mechanism by which BPA actually attaches to a protein. I was taught in my undergrad, you cannot have an antibody to something that isn't a protein. So how are we making antibodies to BPA? Well, they have shown in this particular study that that chemicals can bind to our own proteins like albumin and form a completely new antigen. Since the protein binds to self, the antibody formed to the new antigen can actually result in an attack of self. So chemical immune reactivity does not depend on a load. It depends on exposure, loss of tolerance. Improving tolerance is more important than trying to avoid the chemical, because that's nearly impossible. This particular diagram out of that very same study, I've highlighted here, if you're using the 3x4 blueprint, this kind of speaks to, the little purple circle speaks to our detoxification, what is happening in detoxification, what is happening in inflammation, what is happening in hormone balance, as well as our immune, histamine, vitamin D pathways. But Annelie is going to take you through the genetics. This I found incredibly interesting. The alkalinity in water is such that it reduces the chances that BPA can even be leached into water unless it's heated. However, common fizzy drinks have a much lower pH, making them very acidic. And as a result, BPA can be leached out of the bottles that these cold drinks are held in. Something to think about. So is it a failed immunity that resulted in the development of autoimmune conditions, or is it the introduction to our environment and diet substances that have the potential to mimic our own body tissue if they enter through a barrier which has lost integrity or been damaged by those very things that were meant to help us? I'm now going to hand over to Annelie to speak to the genetic component of autoimmunity. Over to you, Annelie.
Thanks, Nikki. Okay, so that was very interesting. Thank you very much. Okay, so there is my screen. So I'm going to take you through some...
There we go.
Sorry, yes. So I'm going to take you through some genetics, and as Nikki said, we will be here all day, but I'm going to try and highlight some of the most important things that you'll come across, and starting off with also just my introductory slide. So we know that autoimmunity can be associated with any tissue or organ, and Nikki spoke about the environment and hormones and immune system, and so I'm going to cover the genetics, but this is a great slide, and I often encourage you, if you have a practice eventually, to save these slides because it's really engaging with your patients. So as Nikki mentioned, this autoimmunity is this runaway response that results in the body attacking itself. And so when the immune system becomes confused and your own tissues are caught under crossfire, like she said, conventional medicine accepts the problem but address it only by patching the symptoms. So what we want to do, looking at genetics, we go upstream and we want to know why the body is out of balance. And that is just an important piece of the puzzle. Of course, we don't ever only look at genetics, but we look at this puzzle. So I'm going to just go through this tool with you. And so starting here, so basically from a functional medicine's perspective, we want to obviously consider all the aspects that play a role in the progression up to the disease. So if you look closer at this tool, you'll see we're trying to make this link where we are creating the link between environment and genetics. So that will help us to just treat each person individually. So if you've ever treated autoimmunity or you experienced with it, you know that every person is different, although it often presents the same. So if we start with the triggers over here, so these are the environmental triggers that Nikki went through, and then these are the genetic. So these are the pathways in the 3x4 genetic report, and we will definitely look at how you respond to these triggers would also be impacted by these pathways. So I will go through them in more detail, but these are the pathways where we have groups of genes that impact them. And then moving on to the immune dysregulation, these are more of the pathways. So the cellular defense mechanisms would be these 4: detox, oxidative stress, inflammation, methylation. But then we also have the hormone balance, the vitamin D, HLA are your human leukocyte antigen genes, fatty acids and histamine. And then you have this loss of self-tolerance, the production of the self-tissue antibodies that Nikki explained so well. Then you have this silent autoimmunity where you actually have the low NF-kappa beta, which we'll talk about a little bit more. So that's a transcription factor that impacts your immune response. Your high TH3 and low TH7. Again, the cellular pathway is playing a role here. And then moving on to autoimmune reactivity. Again, the genetics playing a role, and then autoimmune disease. And then also just thinking about the gut. So the gut plays a huge part because this loss of integrity that Nikki spoke about just now is caused by these triggers and dysregulation, and that then causes intestinal inflammation as well as nutrient malabsorption. So remember, we need these nutrients to help activate certain processes and genes. So you have the inflammation, but also the malabsorption and the immune response that contributes to this multiple food intolerances, which was very interesting. Thanks, Nikki. And then the autoimmune diseases. So remember also that autoimmune diseases will also target tissues, which can also be impacted by genes. For instance, if you have gene variants in connective tissue, you'll often find that these are the target tissues. But just a little closer look at the gut. So the role in autoimmune disease, we know that it's very important if you treat autoimmune disease that you're going to start with the gut, but where do we start? So that's you often hear, oh, you must support the gut, but well, how are you going to support the gut? So of course, the microbiome will impact the gut function. But remembering that the gut is the largest immune organ in the body, it's important, obviously, for immune balance. But what's also important is to remember this epithelial tissue or epithelial cells that line the gut. So they will be impacted by these specific cellular functions, which I'm going to go through. And that's really where you're going to start. You're going to see how are these cellular functions impacted, and remembering that if you address those, you are going to be supporting this gut barrier. Right, so what are these cellular functions? So they all affect each other. So this is just the introductory slide. So you'll see they all affect each other, but starting at the left here with methylation. So we get biochemical methylation as well as DNA methylation, which is where genes are silenced and it's impacted by epigenetics. I'm not going to talk about that. I'm going to talk about biochemical methylation, which is the attachment of methyl groups to substrates like toxins as well as other endogenous substrates like hormones. Then we have our oxidative stress pathway. So that's the redox status, which can also be sometimes referred to as the pH balance. And that is also where we have this balance between the production of reactive and oxygen and nitrogen species and the cleaning up of that. Our inflammation pathway, which will also impact membrane integrity, endothelial function, vas circulation, and vasodilation. Sorry, all of these pathways will impact that, of course, because of the lining of all these places with these epithelial tissues, and endothelial tissues. And then we have all of these will also impact energy production. And lastly, we have our detoxification processes, which mainly happens in the liver, but every cell also has to be supporting your detoxification processes. Okay, so let's look at inflammation. So that is obviously a normal physiological defense. I'm not going to go into more. Nikki explained it quite well. But of course, if we have chronic inflammation, that will be explained by genetic variation, and that will be associated with autoimmune disease. And you can also have an over-responsive adaptive immune response, which is associated with autoimmune disease. So these are some of the genes. Of course, there are many, but the idea is to look at this diagram, which just explains the dynamics of the inflammatory response. So you have your acute inflammatory response that is coordinated by a sequence of events, which was very well explained just now. And this is the coordination of the chemokines, the cytokines, the pro-inflammatory mediators. So genes that will impact how these behave are specifically genes like TNF-alpha, interleukin-6. Interleukin-6R is the receptors where interleukin-6 attaches, as well as interleukin-1, and they often inherited in groups. We call it a haplotype. And if you have gene variants that impact these, often they're associated with with an increased pro-inflammatory response, and that it actually carries on for longer. Then you have your resolving phase. So remember, just using anti-inflammatories are not going to help always to resolve the phase. So these are the different processes that happen. So we have, for instance, our FOXO3 gene. So FOXO3 is a transcription factor, which means that it activates genes in the cell to help to resolve the phase. So it activates many genes. For instance, it activates things like interleukin-10 as well as apoptosis. And so that is one of the genes that we look at in terms of how we resolve this phase. And then also looking at genes in the gut, like these three here. The FUT2 gene is called the fucosyl-transferase enzyme, which is important for the production of H antigens or the secretion of H antigens that you also find in blood groups, and that impacts bacterial balance. You have your DAO, for instance, which is your diamine oxidase enzyme that impacts the breakdown of histamine, which can also cause inflammatory responses. And of course, your HLA genes, which are the human leukocyte antigens. So all of these will impact the inflammation, and if you have a genetic test available, you'll be able to see which of these genes your patient has inherited. Then looking at oxidative stress, so that also occurs in autoimmune diseases, and that's, like I said, the imbalance between your reactive oxygen species and nitrogen species generation and their removal. So it's catalyzed in the mitochondria and the cells by various enzymes that is listed here in the green block boxes. And then your antioxidant enzymes are down here in the pink boxes. So that's things like superoxide dismutase, catalase, glutathione peroxidase, and there are gene variants that impact how these antioxidants are actually behaving. So what's also interesting is that you have these native proteins that can be damaged by oxidative stress, and they are called neoepitopes, and they also are associated with autoimmune diseases, specifically these listed here. And they are basically also seen as something that's foreign. So this protein is damaged, and this is something the immune system has not encountered before and evokes this vigorous T cell response. So now we all have this image of how a T cell works in our heads, hopefully. So oxidative stress and inflammation, there's this vicious cycle where oxidative stress and inflammation just carries on in a cycle. So when signals are detected, and that's normally signals like viruses, bacteria, stress, and also just increased production of these reactive oxygen species in normal metabolism, you will have the damage that takes place, but that is through the activation of NF-kappa beta. So NF-kappa beta is a transcription factor that activates the immune system. And then you have this increased expression of these pro-inflammatory genes. And remember, you can have gene variants here where they are over-responsive. And this then causes tissue damage, which causes production of more of these reactive oxygen species. And that then signals again that's recognized by the immune system, and the signal, this actually continues. So if you have an increase sort of interaction between your inflammatory gene variants and oxidative stress, that also increases the potential for for autoimmune disease. So this is just a little bit more information. So I'm not going to go into more details, but what I want to show you is these are the genes. If you do the 3x4 test, these are the SNPs that you can test. So these are the oxidative stress genes. So if you have SNPs in these, in other words, they have a reduced capacity to quench these oxygen species and nitrogen species, you can treat it by using cofactors, which helps to upregulate these genes. And you can also upregulate NRF2, which is a transcription factor that helps to upregulate all the antioxidant systems in the body. Right, detoxification. So, of course, you get exogenous and endogenous toxins that can also trigger autoimmune disease. So in your gut, your gut is really important because that's going to remove the toxins, and that play a very important role here. If you have a slow detoxification process in the cells which are impacted by genes, there's a longer chance for these toxins to attach to a protein, which is that mimicry that Nikki spoke about, and that can then increase your possibility of autoimmune disease. So this is quite a busy slide, but it's just to show you how the detoxification works, the pathways works. So again, the toxins will damage the tissues the same way as which I described the oxidative stress just now. So these chemical structures, like the ones in oxidative stress, the body responds to that, increases the inflammation, and you have these autoimmune responses. So the 3 phases. So the first one is so the toxins will enter the body. Remember, you can have a breach of these barriers, enter the cells, but also, of course, these are waste products that are produced in the cells themselves. So your first phase is mediated through your cytochrome P450 enzymes systems, and this converts all these chemicals into more toxic metabolites, because now it's being prepared for phase 2, where it conjugates to toxins and prepare them for elimination, which are more water soluble. Then you also have your phase 3 that's often not always talked about in the literature, but phase 3 is critical because it involves the transport and elimination of these structures through cellular membranes, and this also includes the blood-brain barrier. This is, I'm not going to go through these slides, but I thought I'll just include them for you to look at afterwards. These are all the gene variants that is in phase 1 that we test, and this gives you a good idea of what chemicals they interact with. And these are the phase 2 gene variants, all the processes for conjugation of these toxins, and all the genes that play a role there. Then with methylation, so methylation, of course, is carbon metabolism, and it happens in every cell. It's important for DNA repair, but also helps with the regulation of detoxification, oxidative stress. So this is really important for the epigenetic programming of your CD4 and your T helper cells. And it will also influence the expansion of your pathogenic Th17 cells. So again, if you have genes that impact this process and you support that, that's really going to help support your patient. This is just the transsulfuration pathway that I mentioned. So if there's oxidative stress, it will impact the formation of glutathione, and also the formation of taurine and sulfate, which is important for detoxification. So all these pathways interact with each other. You also have system pathways. So there's many, but these are the three that stand out. So your hormone balance, your glucose insulin. In our glucose insulin pathway, we also look at the deiodinase 2 gene variant, which impacts the conversion of T4 to T3 in the thyroid. So if somebody's got Hashimoto's, that's quite an important gene to look for. And then also your histamine that impacts your inflammatory response. So now starting with hormones. So Nikki's talked a lot about hormones. So remember that estrogen are enhancers of your humoral or adaptive immunity, and your androgens, as well as progesterone, is natural immune suppressors. So whatever is going on in your hormone pathway, which can be impacted by genes, will also help. If you support that and get the hormone balance right, that will also help with the treatment. Yeah, so, and we also know, and I'm not going to go into more detail because Nikki explained the role that hormones play, but we know that autoimmunity is definitely found more often in women than in men. Then histamine. So these are all the signs of histamine overload. Histamine overload is if there's an increased production of histamine, either from the gut or self-production. There's all sorts of reasons we have high histamine, and also the breakdowns of histamine, where you have the gene variants that impacts the deamine oxidase and histamine methyltransferase enzymes that is like blockages in this drain. So you have an increase of histamine. So this is a great tool that shows on the right-hand side the triggers, so your genetics, where you have reduced function of these enzymes, along with whatever happens in your gut, histamines in food and so on, that are triggers. Then your oxidative stress detox, also triggering histamine release, and that causing inflammation, histamine attaching to these receptors, and having lots of different responses leading to these symptoms. But there is some evidence that the histamine attaching to your histamine 4 receptor specifically, that are associated with specific autoimmune conditions such as Parkinson's and osteoarthritis. And just think about taking antihistamines, which blocks your H1 receptors, or your proton pump inhibitors that can block your H2. The histamine is still being produced. You're just blocking these receptors. It's driven down and attaches to these other receptors, and that can have an increased response here. Then nutrient pathways, vitamin D, gluten, B12, and fatty acids. So vitamin D, also a very busy slide, but just so you know, vitamin D activation. So there's genes that impact how your vitamin D is activated in the liver and kidneys to form your 1,25-dihydroxyvitamin, and that interacts with vitamin D receptors, which are also transcription factors that are really important for the activities of the innate and adaptive immune system, and also which suppresses inflammation. So it also impacts NF-kappa beta. So the role of vitamin D is very well known in autoimmunity. And again, you can test somebody's vitamin D levels, but it doesn't tell you what's going on with their vitamin D receptors and the activation. So again, where gene testing is really valuable. Then vitamin B12 and fatty acids. So B12 is impacted by gut health, and we also have conditions like pernicious anemia, which is an autoimmune condition that further impacts vitamin B12 levels. And then fatty acid pathways, where we look at how fatty acids impact how fatty acids are elongated in the body, and some people have genes that impact the ability to form your EPA and DHA, which has got anti-inflammatory effects. So in that case, the diet and the fatty acid balance in the diet is very important. And then, of course, our gluten pathway. So in our report, we call it the gluten pathway, but it's really the pathway that helps us see if somebody has the HLA genes that's associated with celiac disease. So these are the symptoms of celiac disease. It's often missed. It can happen at any age. And instead of going and doing biopsies, you can actually first screen for the genes to see if somebody's got a chance to have celiac disease. So this is the major histocompatibility complex class 1 DQ genes. You don't have to remember all of that, but these are genes that are inherited in a haplotype, which means you inherit a group of genes together. And some gene tests talk about DQ2, and they then diagnose celiac. but it's actually the specific gene variant, which we call 2.2, 2.5, and 8 that you inherit. So what you need to know is that if you have the DQ 2.5, especially homozygous, that you have the highest risk for celiac disease. And that has got to do with the probability to have tissue damage because of the way these gene variants impact the cellular tissue. So it basically plays a role in how the antigenic peptides present on the cell surface and how it's recognized by CD4 and T cells. That's a whole lecture by itself. But what you need to know is if you have a positive result for DQ 2.5 or DQ 8 variants, you can then look for further investigations instead of doing it the other way around. Because if you do this first, and you don't have the HLA genes, you might have a non-celiac gluten sensitivity. So just a little picture here. So you could look at the genetics that impacts the innate immune system. You have the production of antibodies and the deamination of gluten peptides. You have the TG2 release, and then that all impacts the cells, the villi in the gut. Sorry, I'm rushing here because I know we're almost out of time. Okay, so let's go through our case study. I've got a case study here of somebody who had multiple autoimmune diseases. And what I just want to demonstrate is how you will interpret genetic information to support the treatment. So this was a patient who at age 59 had many different autoimmune diseases. She was diagnosed with ankylosing spondylitis, and that presented when she was 15 years old. She had reactive arthritis from an infection when she traveled in Egypt when she was 26, and she was immobile for 6 months, and it took 12 months for her to recover. Psoriasis was diagnosed at 30, and arthritis age 40, Crohn's disease over that time as well. In 2006, she also has this HLA-B27, which we don't test, but it's associated with ankylosing spondylitis. In 2006, she was severely overweight. She was 2.5 times her current weight when I saw her at the time, and a realistic weight for her was, I've got it in kilograms, but she was about twice her body weight. And she wasn't on any medications at the time. She was desperate, went onto medication, and everything got worse. And she had terrible gut pain. She also had PCO, and also these were the viral infections she had. She had a family history of autoimmune disease, and her mom had lymphoma, but in remission, and also diabetes. So she wanted to treat her autoimmune condition, reduce her pain and inflammation, manage her body weight, and also expand her dietary choices. When I saw her at the time, there was very little that she was wanting to eat because she was just reacting to everything. So we wanted to heal her gut as well. In terms of lifestyle, she was on many elimination diets. She wasn't eating grains, lectins, dairy, nightshades, eggs, and legumes. She wasn't taking any alcohol, and also loved exercise, but wasn't able to do it often because of pain. So she was already doing a lot, but really was at her wit's end. So I want to show you, I don't know how many of you have seen our genetic report, but the purple areas represent high genetic impact. So this is probably one of the most purple reports I've seen. She had high impact in almost every single pathway in her report. And I highlighted the ones here. So you can see she had gene variants, very high impact gene variants in all of these cellular pathways, as well as actually all of her system pathways. So that is one of the reasons, besides everything else that was going on, she was really struggling. So what to do with her? So in terms of triggers, she had all of these hormones, B12, you name it. In terms of dysregulation, she had vitamin D. She didn't have the HLA genes, but she did have fatty acids and histamine. And also, again, for the silent autoimmunity, which happened earlier in life, she had all those cellular pathways, as well as the ones that increased her actual development of the reactivity stage, and also the genes. All of these would have impacted her gut health. So we really focused on gut healing. So this is a picture, just again going into detail of the inflammation and methylation pathways. At the top, I just mentioned the most impactful genes, and all of these would have contributed to this chronic inflammation, The DIO gene here impacts histamine production, C-reactive protein, and also the FUT2 gene here that's associated with gut inflammation. With methylation, she had this slow functioning COMT, so a slower methylation, and also impacted by stress. So if she's stressed, also more of these catecholamines to be broken down. The MTHFR gene needing more folate and MTHFR that impacts methylation, and needing vitamin B12 as a cofactor, and a B12 absorption rate was impacted, which we could see for the genes, and also she had a high impact in a B12 pathway, so needing more vitamin B12. This was a systems pathway, so again, hormonal balance, hormones playing a role, and she did say that she had throughout her life when she had hormonal changes, especially when she was older, going through menopause, causing problems again, and then a histamine overload. Estrogen also activate reduction of histamine. If your progesterone is low, you also don't get the, your DIO is also down-regulated. So histamine also playing a role. This was something she had not thought about before. So we could also look at her histamine in her diet, and also increase foods that helps to break down histamine. Then in her nutrients overview, she had increased need for folate, she had high impact in vitamin B12, vitamin C, and very high vitamin D. So very important for her to look at that. Gluten was still important, even though she didn't have the HLA gene for celiac disease. And because of all her autoimmune conditions, we still decided to exclude gluten. So these were the genes. She had 2 VDR gene variants, the CYP2R1, which means she doesn't activate vitamin D. She also needs to cut out caffeine, because that would make things worse. Also because of her high impact in the detox pathway, and the B12 I've already mentioned. Also, just for interest's sake, she had also all these pro-inflammatory genes that increases the retention of fat cells. So remember, she was very overweight at the time, and that was also associated with the inflammation that she had. So what do? We want to support the high impact genetic cellular system. So you won't normally get a report with so much purple, but we really wanted to think of what we were going to do with her. We couldn't treat everything at once. We want to look at inflammation, reducing that helping her to start with exercise, see if there's infections that she has that we need to test for, stress management, sleep, and also managing her toxin exposures. So what we try to do, just to make sense of it all, we try to look at 3 metabolic pathways to focus on. But with her, I actually took her cellular pathways as one, because they all affected each other. So we'll look at that to help reduce inflammation. We wanted to support a home hormone balance, which we'll also do through detox and supporting these pathways, and also connective tissue support, which is where she had a lot of symptoms. So nutrients, we want to encourage food that helps with detoxification, ensure intake of minerals to help upregulate your antioxidants, eat foods rich in polyphenols to reduce inflammation and oxidative stress, and support the methylation through the optimal intake of these B vitamin foods, and choline and folate. We want to reduce or limit histamine foods that's high, any toxin exposures through processed foods, as well as caffeine and alcohol. And then her lifestyle changes start with some moderate exercise to modulate inflammation. And we also wanted to support a glucose insulin balance, because she had genetic variation there. We could start with things like sauna, massage, sweating to help with detox, and really look at how we can support sleep. Intermittent fasting could also help to support her immune regulation. And then really look at the environmental toxins, those endocrine disruptors Nikki talked about, and what her stressful exposures are. So we implemented a lot of these and worked on the gut barrier. And I saw again 3 months later, she had said that her psoriasis that she was really bad at the time had cleared up over almost 90%. She was very excited to be eating in restaurants again. She wasn't reacting to everything anymore. She was pain-free when I saw her. Remember, she'd already done a lot, so we really just wanted to do some more things. She was able to move and look after her grandson. She wasn't on any medication. We were treating her on supplements, and her energy levels were improving. Right, so that's my one. And so just a summary, when you use genetic testing as a screening tool, it will really give you insights to formulate what further testing you want to do, what targeted support you want in terms of upstream functions, what diet, nutritional and supplement support you want, as well as targeted lifestyle changes, just to help you focus on those and not feeling lost and wondering where you want to start. Okay, so that is my final slide. Okay, we have gone a little bit over. I'm hoping that some of you can stay for some questions, and if there are any, and hopefully, and it is recorded so you can watch it later. Okay, well, there is a question. Can you expand on how genetics play a role in premature ovarian failure? We know it runs in families. And I have had patients like that where we looked at the genes that we test, and I actually can say that we don't actually, I don't know how those genes are connected. Do you know, Nikki?
No. So, no, I don't. I think also what we need to remember is the kind of genetic testing that 3x4 does is low penetrance, not diagnostic per se. So we would use that to give us insight into how else to help manage. So looking at her hormone balance, looking at cellular processes, detoxification, I think that's how I would look at that.
Yes, so that's exactly. So we don't always know the mechanism. There are lots of higher penetrance genes, and often nobody really knows which genes are causing it. We know it runs in the families. What we are interested in is how are we going to support it? How are we going to look at these systems and supporting your patient instead of trying to figure out what gene was caused the condition in the first place? Because we already know you've got the condition, if that makes sense. We just want to see how we're going to support it. Yeah.
I get a minute. Yeah.
I think there's a question about where will we receive a link to the presentation? Yes, so you will definitely get it through your through your restorative wellness.
Yeah, we'll give it to Margaret and she can make it available. Yeah. And then there is a question about IgG4 autoimmunity. I'm not sure that I've heard about IgG4, but if you're talking about IgG testing, what I often find is helpful is the IgG testing when you've got numerous foods. It really gives you an indication that the gut barrier is a disaster. So it's not a diagnostic of you're allergic to those foods, but it is a huge insight. And what I find is helpful is do the gut work and do it again, and you'll see that those IgG numbers either go down or they go away completely. And now you get a better picture of actually what the patient genuinely is so-called allergic to. Remember, IgG is not considered an allergy marker, but like I said, I view it as a gut permeability marker.
Yeah. So instead of trying to cut out everything that you think they're allergic to, is start with the treatment upstream, and then you can test again. Yes.
Exactly. Awesome. All righty. Thank you, everybody, for your attention and your time. I hope you've enjoyed it as much as we have enjoyed sharing it with you. And yes, the link obviously and recording and all of that will be in Margaret's capable hands through Restorative Wellness.
Great. Thanks, everybody. Have a lovely day.
Everybody. Have a wonderful day.